Hi, I'm Dongyoung Kim, an undergraduate student at Zoology University. My presentation topic is the integrative taxonomy on genus Kaolinonychus, Korean and cryptic harvestmen. It is well known why cryptic diversity is important and how it contributes to ecology, evolutionary biology, and conservative biology. Caves and subterranean organisms harbor many cryptic diversities due to the combination of morphological convergence and geographic isolation. Genus Kaolinonychus I studied is a member of Parachidae, which has high regional endemism, low vegetativity, and strong ecological constraints. In addition, according to previous studies in another member of Parachidae, morphology in reproductive organs does not sufficiently reflect their high genetic diversity. Genus Kaolinonychus is an endemic harvestman found throughout South Korea, and the genital morphology among them is also very conservative. For this reason, only a single species with a broad distribution was described under the traditional alpha taxonomic system. However, when observing them, they look different somehow, but it is difficult to figure out which part is different and how to set qualitative criteria. How should we approach this cryptic diversity? For this, an integrative taxonomic approach was used for deeper investigation using the study of Durkheim and Hedin in 2014 as a reference. I conducted the analysis using mitochondrial C1 gene sequence and morphometrics data with 65 length variables. Here is the result. First, in the result of phylogenetic analysis, we can see 3 major clades, and in detail, there are 6 clades. I set these 6 subclades as periodic groups in analysis because these clades are identical in both maximum likelihood and Bayes entry. But the inner clade, marked with the types of yellow, show low bootstrap value and posterior probability, which means that they are weakly supported, marked like this. And then I conducted dimensionality reduction of morphometric data. This is a result of PCA. Next, in PAM clustering, 2 to 4 clusters were found to be suitable, and the clustering results for each K were shown as the following. I established the final species hypothesis, and I was able to estimate the 3 major clades to 3 individual species. In the validation process, first of all, pairwise FST was calculated to estimate the degree of genetic differentiation between subclades. As a result, the red clade displayed above 0.75 from all the other clades. The blue clade showed over 0.5, while the 4 yellow type groups showed around 0.4 or below among each other. Next, t-SNE clustering using morphometrics distinguished the 3 clusters as separate groups. And this is PRSD result. The 3 clades are well grouped together. And when you look at the contributions of variables in PRSDA, we can see that the length ratios of walking legs are all ranked high among 65 measurement variables. This result is noteworthy. ABGD and BPTP algorithm were used as molecular species delimitation, both of which distinguished much more species than hypothesis. This seems to be due to the high genetic endemism in Parachidae. And finally, I concluded that these 3 major clades could be determined as separate independent species. This study is thought to have rendered meaningful conclusions in that we discovered the cryptic diversity from the harvestmen that has been hidden in a veil for a long time. And it is expected that the method used in this study can be applied to other cave and subterranean cryptic diversity. Thanks for watching.